So tell me about the center itself. Do you have any prior bioprinting experience?
I do. I was in University of Otago working with Professor Tim Woodfield in the CREATE group. So we were developing different materials for bioprinting.
I see. Which university did you say?
University of Otago.
Oh, Otago. Okay. Is that in
New Zealand.
New Zealand?
Yes.
Oh, okay. I see. Okay. So you have had past bioprinting experience. Which
Yes.
bioprinter did you use?
The bioscaffolder. It was like a custom built printer, which had different printheads where you could print thermoplastics and soft polymeric materials like hydrogels as well.
Okay. I see. But does that include any cell printing as well?
Yes. Yeah.
What type of structures were you printing?
I was there I left Otago in 2018, and that is when we were working in the bioprinting space at that point for 3 years. So we were printing very basic lattice structures using materials like gelatin methacryl oil and stuff, which were hard to print. Mm-hmm. We were trying to use different fillers to improve the rheology and get the materials printed in a way that they could be printed, and comparing how different filler concentrations will be affecting the printability and also the functionality of the scaffold.
Oh, that's good. Good to know that you have prior experience. What about the rest of the audience?
The rest of the this is we are based in dental school, and we do a lot of a mixed kind of, we have a group of people who work in different spaces. So we have people who are clinicians who know the issues that occur in the dental space and where things can be modified. So they bring their kind of expertise. We do a lot of mechanical testings of different commercial materials and figure out and do mathematical modeling and stuff like that to figure out why those materials fail if they fail. And then I was working with Professor Conrado Aparicio, who is moving back to Spain in May. And with him, we have been mostly focusing on surface engineering for improving the performance of different implants, whether it's soft tissue regeneration or hard tissue regeneration and things like that. So like Wandy over here, he does a lot of mathematical modeling and comparing that with the mechanical data. Wandy, please correct me. I have a bit of understanding of what you do, but not a detail. But
Good morning. I'm Wandy. I'm working on mathematical modeling to predict the shrinkage stress of composite materials. Yeah, so I'm using the finite element approach.
Does the division have any bioprinter right now?
Or no, we don't. But I collaborate with a group which has a bioprinter. We have like a Cellink bioprinter with 6 printheads.
I see. Within the university campus?
Yes, it is.
Okay.
So it's with Dr. Jacob Toller's lab that I collaborate with. Yeah. They had recently purchased, not recently, they purchased it quite a while, but they just got it delivered end of last year and got it installed and everything this year. So they are highly focused in RDEB, which is the epidermis bullosa disease, which happens in the skin, which is a butterfly effect, where the epidermis doesn't attach to the dermis, and it's quite fatal. And the patients are like very young who are born with these defects and they don't survive. So they use a lot of cell therapy based approach to improve the stem cell therapy approach to improve their life expectancy and to treat the condition. And they were interested in doing that because I have a bit of polymeric experience. I've worked with hydrogels. I know how you can modify bits and pieces here and there. So we started collaborating on that project now.
So I'm assuming then in the audience, we do have a mix of people who have no prior experience, because I do have a lot of introductory stuff in my talk. So I'm trying to assess if I should go over those or we simply skip.
No, I think it would be a good refresher for me as well. It's always good to go back to the basics and understand them well. So I was trying to get more information about your particular set of printers. So how is this different than any other printer that's out there?
Okay. All right. Let me first. I think that's a good question you asked. So this is a quick look at. Okay. That's the Dr. In Vivo 46, which is our newest bioprinter that was launched last year.
Okay.
And let me go to the specs. It's also a good thing to start here. This is Dr. In Vivo 4D2, which is a dual module, which means you can use 2 print heads at a time.
Okay.
This is previous generation, but it still continues to be available. And then based on, okay, again, just to go over this, it has syringe dispensers, it has filament extruders for all your filament extrusion, and then we've got the pneumatic hot melt dispenser to handle pellets, powders, and paste. Some of these materials, which I'm pretty sure you're quite aware of.
Yeah, but I'm just trying to understand what's the difference between your filament extruder and your dispenser? Because
Yes.
with the bioscaffolder that I've worked in the past, I could still use the pellets in the extrusion head and you could heat it up and that is how it would melt and that would come up, that would be still extruded as a filament.
Yes,
So,
ours is an extrusion printer as well. Now, the syringe dispenser is only for the soft hydrogel materials. The filament extruder, the input material has to be in the form of a filament. So that's more like your FDM. The hot melt is where you can put pellets, powders, paste, and one standout feature for our hot melt is that we have the highest temperature and pressure ranges available in a comparable competitor equipment.
Okay.
So it goes as high as 350 degrees and then close to 9 bars or 10 bars of pressure.
Okay.
And the nozzle sizes range from 80 microns going up to 400 or so, which is, and you can pick whichever one fits your specific application, but the resolution is pretty high. And also, as an improvement based on our ultimate goal of being able to create an organ regenerator, we realized that for in situ printing, the printer should be able to do curve printing.
Yep.
And also there is a absolute need to incorporate the sterilization modules and incubation functions, kind of build in a bioreactor into this if you want to take that route of making this a truly clinical grade organ regenerator. And so out of that idea was born piecing together of all these existing elements. And then we have the In Vivo 46, which was launched. And 46 has 6 print heads, 5 of them on a rotary turret, and then the others are fixed, especially the hot melt and filament extruders. And so obviously this becomes an all-in-one solution, able to handle complex architecture and high resolution for various tissues, be it the retina or bone, hard material like bone, or beating tissues like heart or muscle.
Okay.
And then I did mention about the integration of the sterilization functions and the HEPA filters and the ability to control temperature, humidity in the printing. As you can see from the name, we don't like to call it a 3D bioprinter. We call it a 4D because we do take into account the time dimension there and the need for cells to mature to become fully functional. So to make that happen, it is important to make sure the cells are happy all through the process. Sorry, you were about to ask me something?
Yeah, because for me, 4D printing, I've read a bit about 4D printing, where an external stimuli can cause a change in the structure altogether. So I was assuming that you are designing some specific inks for that. that is not correct. You do?
We do have, basically ROKIT is a well-rounded regenerative medicine company, more than just a bioprinter manufacturer. So they do have dedicated bio ink development. And notably, we have a product called the Humatine, which is cell-specific native ECM harvested in its native form without any denaturing. So you get cell-specific, let's say you want cardiomyocyte-specific extracellular matrix-based bioink, then it's possible. They do have a proprietary cell sheet technology using which they harvest the entire gamut of ECMs, not a single component ECM. And so that is very relevant, especially when you are pursuing a clinical product development. And no other single component ECM or bioink can even come close to what that can do. So they do have all those offerings. I mean,
okay, just a very dumb question here. So would you be modifying that ECM to make it cross-link better so that it doesn't denature, or not denature, but destabilize over time? The reason why I'm asking is because I am working with someone who just mentioned to me a few weeks ago that they had issues. So they were doing something similar. They were trying to derivatize ECM from bone and use that in combination with other materials. And she was telling me that in vivo, the material is not stable at all. Like in vitro, the system is stable. You can handle it over 3 weeks or 4 weeks time. But in vivo, the whole system degrades in less than a week.
Which material did she use?
They were derivatizing their own material.
And how did they harvest? Any idea?
No idea.
No. Okay.
No.
Daye is our bioprinting specialist from the manufacturer side, ROKIT Healthcare. Do you have any insights on this question, Daye?
Hey Isha, this is Daye from ROKIT Healthcare. In response to your question, it may be best for me to refer this to our Humatine product director, but as far as I know, we do not intend on modifying or adding any additives or whatsoever to modify the ECM that has been extracted wholly from the cells. So the point is on basically creating, native ECM derived from cell-specific or tissue-specific sources that could be potentially be clinically applicable because we are basically deriving these from human primary cells instead of animal sources. So yeah, for applications that require translations to medicine, this is where we are this is where the issue is that we are trying to address. Like, for example, if Metrology has been a material that has, successfully advanced research in 3D cell culture, but it still has that remnant challenge of not being clinically applicable.
Yes.
But yeah, it would be interesting though to, to replicate the research that the group has done, where they've seen that ECM derived with their method from bone didn't last as long in vivo as in vitro. So I could definitely talk to the product director and the research scientist to see what they have seen before.
Okay. So just to clarify, when you say they're derived from primary cells, so you're not deriving it from tissues, you're doing it differently? Or did I interpret it all wrong?
Oh, no, you're correct. This is this technology called cell sheet engineering.
Oh, okay.
Depends on basically the growth and proliferation of cells, human primary cells, on a like an environmental stimuli responsive polymer bed. And by basically modifying the polymer in terms of ion stiffness, you could also sort of modulate, the type of mechanical environment that these cells live on too. So you can both vary the cell-specific source as well as the mechanical environment of the bed on which the cells live in order to produce their natural ECM.
Okay.
And then once the ECM has been produced, we have the technology of basically extracting the ECM without or by minimizing denaturation.
Okay.
And then we basically cryonize the ECM and can make them available either as supplements to bioinks, like naked bioinks,
Yep.
or provide them for 3D like cell coding, as well as bioinks themselves.
Okay. Would you mind sharing that information via an email? Because I might be interested in something like that. I think I know why they have issues, because they're not doing what you guys actually do to make that derivatized ECM. They're actually derivatizing it from a tissue. So decellularizing the tissue and then getting it. I think that is where the platform varies significantly.
Okay, yeah, I can definitely share more information about that through Sensofar. But we also do have what's called Dr. In Vivo Cell, which is another development that basically allows you to decellularize and recellularize at the same time from tissues or from organs. So I can send you more information about that as well. So you basically have products from two different concepts.
Yeah,
but they all support tissue engineering, regenerative medicine. But just to add to the point about 4D bioprinting, I completely agree with you that the concept of 4D printing is definitely what you have mentioned. And I think the world of 3D printing as it developed into 4D printing has definitely focused its attention on the responsiveness of the biomaterials to external stimuli, such as time. But we would like to put a new spin on the 4D bioprinting by basically saying that in the biological world, we're always dealing with moving cells that are not just moving in time, but also, functionally. They are always in motion, in dynamics. But when these cells are exposed to environments that they are not native to, and which could be, something that as simple as on the bench or inside a bioprinting chamber or through tiny nozzle tips of the printhead, what can we do to best preserve the native environments that they exist in, so that with time or with their normal functions, they would be able to retain their sort of characteristics. So with that, we have started putting more focus on the hardware as well, not just the biomaterials, but the hardware. And we have included the bioreactor as well as a sterilizer, so that printing is not just about printing of the cells, but it's about basically optimizing the conditions in which these cells are exposed to before, during, and after printing. So that's what Sentil and I would like to emphasize in our slides. And you will see later on, as he starts the presentation again from the start, that we are a company that for the first time has placed the bioprinter in the operating theater. So he'll talk about more about that. But within that journey of placing bioprinters in the operating theater and really talking with doctors and healthcare professionals around the world, we have learned that there are certain things that we definitely have to consider that are different from the research scale and translate them to the medical scale. We need to consider certain factors that allow doctors to be basically more amenable to this technology in translating its benefits to patient life. So we will talk about that. And the concept we wanted to stress is we ultimately want to almost develop a mobile GMP inside a bioprinter. So that's something we will address throughout the presentation.
No, that's pretty cool. Looking forward to knowing more details about your cell.
Sure. Now that I started, I'll at least just finish this section so we don't have to go back again.
Yep.
So about the 4D6, it also has incorporated a microscope, and you have the option of a plasma sterilizer, medical grade, if you want pretty high-end CGMP applications to be developed out of this. And it doubles up as a high throughput dispensing system as well for any of these high throughput demanding applications, say drug development or vaccine research or genotyping. And as I mentioned, it can handle diverse materials because of the hybrid technologies that come with its rotors. The dispensing rotors on the rotary module, as well as the extruder and hot melting. And as I mentioned before, we have one of the highest temperature and pressure ranges that's available out there in this segment of the market, as high as 350 degrees from room temperature for the pneumatic hot melt. So it kind of ensures that you have the ability to deal with many different materials, some of which you may not even have encountered at this point of time. So it's kind of future-proof that way as you embark on more innovations. And this is going to be a good companion to fast track to any medical commercialization because of the CGMP readiness. And as I just mentioned, and I'll touch upon this later as well, this is the first bioprinter that's actually being used in the patient care operating theater setting. At least one indication where we have successfully shown that's possible is the diabetic foot ulcer, being able to print custom skin patches for patients. And it comes with a very intuitive and good user interface that can be accessed right on the device, or on a PC, or on a mobile device, or a tablet. So it's very much in line with the needs of the modern networking abilities. All right, so I guess then let me skip to the first half of my presentation, and then we can get started.
Sure. Do you mind giving highlighting what materials have? So this is what you said has already been used in clinic, right, for the diabetic foot ulcer. So
yes.
What material?
The 4D system. Yeah.
Yes. So what material was used?
Okay, the ink itself is derived from the patient's adipose derived material, mixed with adipose derived stem cells. Am I correct, Dai? Yeah, so these are aspirated from the patient's adipose tissue, and then mixed with some let me see, I think some humatine based material. I should be able to get to that.
I'm sorry for asking these questions, because this is something that I'm really interested in.
Yeah, sure.
So,
I mean, they have successful
knowing what,
trial. I'll
So
hold it back till we get to that slide. Okay.
Okay, sure. Sorry. Go ahead.
No problem. All right. So then, can we get started and move on?
Yep. Hey, Senthil. When we get to the DFU slide, I will explain a little bit more.
Oh, perfect.
But
Okay, let's go on.
Yeah.
Okay. All right. Okay. Perfect. Okay. Again, a very good morning to you all. My name is Senthil. I'm the founder and CEO for ONS Bio LLC, which is the Midwest distribution partner for ROKIT Healthcare. And I have with me Dai from ROKIT Healthcare, senior bioprinting specialist, and my colleague Chris Kamimura from Indicate Technologies, which is the West Coast distribution partner. So I guess in the interest of time, I will skip some of these usual introduction, and then let's get started with the introduction, just to make sure it's less distracting. I'll turn off the video. All right. So before I begin, it's important for me to set the context for why bioprinting is important and how it fits into the needs of the current life science research and the medicine, practice of medicine. So first, I'd like to show you this happening that happened in 2018. So if you look at this news is very important for us to recognize. There have been several notable events and mega trends that warrant our closer attention to the growing relevance of bioprinting and its potential as a revolutionary technology for its adoption into biomedical research and eventually treatment of disease. So in September 2018, we saw this news that came in BBC, I guess, where a boy who was living for 14 years with a new urinary bladder made from his own cells, seeded with 3D inkjet machines into decellularized pig bladder as a scaffold. Now, the significance of this work is the fact that, let's see, what's going on here? Okay, the significance of this work is the fact that this work and similar work by many others, it kind of highlights the promise and potential for bioprinting of human tissues to transform medicine with far-reaching implications for organ transplants, cancer treatment, and many other possibilities, such as new ways to develop antibiotics and drugs. And now, the other notable happening is the fact that the American Medical Association approved reimbursement codes for 3D printed surgical models and tools. Now, these are making the foundation for a clinical registry of patient 3D scan data and relevant 3D modeling, which will eventually serve as a learning resource and tutorial. And also, more importantly, it's going to provide the much needed impetus for active implementation of 3D printing in biomedicine. So in September 2019, in the same year, another important happening was the fact that the United States Environmental Protection Agency announced that it'll eliminate all mammal testing by 2035, thus becoming the first federal agency to make a strong statement and commitment to the transition to humane research, while emphasizing the urgency to find alternative solutions to the use of animals in research. So ROKIT Healthcare and a diabetic foot ulcer specialty clinic in India put this bioprinter, which I was talking to you about, in vivo 4D, in the operating room to create a streamlined process for skin regeneration for diabetic foot ulcers. So how this is done, maybe here you can get a little more insight into how this was done. This new and alternative method for chronic wound healing, it promotes skin reconstruction using bioinks that were made from patients' own adipose tissue. And these were printed into size-specific patches, custom made for each wound, and applied directly onto the wounds. These dermal patches, they integrate with the skin, and through early onset healing, ROKIT's mission and vision is to decrease the rate of amputation for patients with diabetic foot ulcer. This is critical because the 5-year survival rate of patients who have undergone amputations is actually far lower than the 5-year survival rate for even cancer patients. So this is an important problem that needs to be addressed. And as you can see, bioprinting has clearly demonstrated its potential. There'll be a little more details towards the end on the process itself. And if I haven't answered your question, then we'll revisit that. So last but not the least, as we all know, in the last year, since the last year, COVID-19 has changed everything about the way we live. And most notably, we have seen that the pandemic is capable of disrupting the traditional centralized value chains in manufacturing. So, for example, when the hospitals got cut off from supplies of masks and ventilators because factories were shut down everywhere and the supply chain was disrupted, 3D printing came to the rescue in some cases to manufacture these supplies as an on-demand, on-site approach. So why am I saying all this? What are these mega trends pointing us to? What's the take-home message from these global events and mega trends in biomedicine? It points to the fact that the 3D printing and bioprinting can react directly to decentralized demands from life sciences research and can also serve as an effective medical strategy. Clearly, there is a growing value for the on-demand manufacturing, biomanufacturing, and bioprinting is a very relevant technique or technology to the real world needs and megatrends. For those of us who might be new to this field in the audience, bioprinting is not new technique or technology. So it's worth paying attention to how this technology is maturing to impact research and healthcare workflows. So the attention on bioprinting is in the mainstream research is just gaining popularity only recently. The origins of the technology itself go back to nearly 2 decades, and it's not totally in its infancy. In fact, the number of research publications around this technology have been growing exponentially since the year 2000. And today there are thousands of publications that serve as a testament to the benefits and applications of the technology across multiple disciplines, tissue engineering, biomaterial development, regenerative medicine, and so on. So when talking about bioprinting, it is very important to point out that this is a platform manufacturing, biomanufacturing technology, which means it has applications for nearly all major funded research fields, as you can see in this slide, be it cardiovascular tissues or musculoskeletal, or for today's talk, craniofacial and dental tissues. It's applicable everywhere uniformly. And this slide shows a lot of hyperlinked publications to demonstrate that how they have been applied in each of these different cell types and organ systems. And I would even stretch this far to say that perhaps in the future, journal publications will start requiring data from bioprinted 3D human tissues as a go-between the 2D in vitro cell culture and in vivo animal or human studies. And it may become an important criteria for your research to move forward and be published. So one of the major challenges, though, is the fact that in medicine, especially, there is a shortage of a number of tissues or organs to replace failing and aging ones in patients. Modern tissue engineering aims to address that directly by replacing the damaged tissue with man-made scaffolds that can support cell growth. Bioprinting as a technology is at the frontier of this field, and because it is the only platform technology that can create these tissues and organs with applications across all these different types of disease that come with aging, there is a lot of evidence actually about what different choices of or combinations of cells and biomaterials can be used for these specific tissue-specific applications involving bioprinting. So the end goal is to be able to use this platform technology and apply it to all these different tissues and disease. So it is, the bottom line is, it is a platform technology and not restricted to any one single area or discipline. And it has the potential to generate or manufacture human tissues for treatment of disease as well. For the ones that, again, who are new to the field, I'll try to define or kind of vaguely define the bioprinting in simpler terms. Bioprinting is a perfect combination of computer science, biology, instrumentation engineering, and material science. It brings the benefits of personalization, autologous solutions. By autologous solutions, I mean use of patient's own cells for the treatment. And it brings fine details and precisions and ability to pattern different tissues at a very microscopic level to improve human health and also advance knowledge of the disease progression and therapies. So 3D bioprinting, all in all, is an additive manufacturing process in which cells, biopolymers, and growth factors are combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time before they become fully functional, there is this inevitable dimension of time involved here. And that's why we believe it's more appropriate to call it as 4D bioprinting instead of just 3D bioprinting, especially when it involves living entities such as cells and tissues. All right, now time to address some basic questions. What are the key components of 3D or 4D bioprinting, however you prefer to call it? Bioprinting technology consists of largely three components: biomaterials, bioprinter, and the design. So the bioprinter represents the hardware component and the design represents the software component. First, let's talk about biomaterials. Unlike the traditional 3D printer, which uses hard plastics, metal, and synthetic materials to create non-living structures, a 4D bioprinter uses materials that are biocompatible, biodegradable, and derived from both natural and synthetic sources. The common examples of naturally derived materials, biomaterials, would include collagen, gelatin, elastin, chitin, which come from the extracellular matrix of human or animal cells. They also include seaweed sources like cellulose or alginate, and sericin, insect sources like sericin, and so on. Natural hydroxyapatite is also a commonly used material, and it comes from mammalian bone. Synthetic biomaterials are biodegradable polymers with both strength as well as elasticity to mimic human tissues. And some of these have been put to good use in FDA-approved medical devices, actually. Finally, you have got the composites, which often combine both the natural and the synthetic materials to add or enhance certain mechanical or chemical properties. Now, these biomaterials can be soft, low viscosity hydrogels, or they can be hard, biodegradable plastics. And when hydrogels are combined with cells for printing, they are called the bioinks. Bioinks basically serve as the extracellular matrix, or ECM, when you are trying to print cells outside the body. Bioinks, they protect these cells from shear stress, and they provide them with nutrients and hydration, and they give the much needed overall physiochemical support to maintain the structural integrity of the tissue that's being printed. So when selecting a biomaterial for use as a bio ink, there are several key criteria, but they can basically be summarized into two broad categories. One, bioinks should be able to extrude out of nozzle tips, stack layer by layer, structurally with or without post-printing processing. Two, the bioinks should be biocompatible and they should uphold high cell viability. Let me show you this video. So now let's talk a little bit about the hardware. Okay, that's just the printing process to give you an idea. So there are various methods and technologies for bioprinting today, as you can see here in this slide. They include the inkjet-based, the laser-assisted, the stereolithography methods. But of all of these, the extrusion is the commonest. More than 70% by one estimate of the bioprinting involves extrusion-based methods. And that's what we are going to focus here today, because our systems are also extrusion-based. One key component of any bioprinter hardware is the printhead itself. And so a printhead should be able to print both hard and soft material. So there are two ways that the printheads can print. One is a scaffold laden way where you can print hard matrix, and the other is the scaffold free way of printing where it does not involve any hard matrix. Another key component is the chamber's ability, as we've been emphasizing while the ability of the chamber to mimic a biosafety cabinet is key, because after all, when you're dealing with living cells, it is important to be able to optimize cell viability and functionality under the stress of printing. The chamber should have UV disinfection capability, and it should have a HEPA filter and some type of bioreactor capability to provide optimal environments for the cells, especially when it comes to temperature and humidity control. Okay, now that we understand the components of bioprinting, we can, the last component I skipped. The last component is the design, as I mentioned before, which is the software derived or software driven design component. But an important one though, because this involves creation of 3D models of the objects that we want to print. The 3D model files, which are known as the STL files often, can be obtained either by manual creation using a design software, or they can be derived from CAD or MRI scans of physical structures like the body parts or organs. Then these STL files get processed with a slicing software that's often inbuilt into the device, as well as available as a standalone software on the computer. What this does is it converts it into standard machine language that describes how the printer should print the job in a given three-dimensional space. It includes the information on the XYZ positioning coordinates, and also provides additional information needed to complete the job, such as printing speeds, infill patterns, etc. And the example, the visualization of these two file patterns is kind of shown here in the bottom, where on the far left you see the STL object and how the G-code variations can be visualized with fine structures. All right, now that we understand this bioprinting process, we can put these things together logically to see how the printing progresses. So it involves 5 key steps. One is preparation, printing, post-printing, evaluation, and application. And the preparation is very intuitive. It's simple. You've got to identify the problem, and then you choose the material, and then the cells are chosen, and then you do the 3D modeling. Then the printing process itself is where you load the material, slice settings and optimization to recreate the structure based on the material properties, and then actually extrude the material and print it. The printer does all of this for you. Then we've got the post-printing process, which may or may not be required depending on what material you use. And that often involves taking the printed structure and doing a mechanical or chemical stimulation so as to create stable and functional structures of the biological material. And that can be done either by thermal cross-linking or chemical cross-linking, or using light photo cross-linking, and so on and so forth. The evaluation is a critical step wherein you compare the cultured or printed tissues and assess for structural, morphological, and functional characterization against chosen living standard tissues to see how well you've been able to replicate and how well the printed tissue will serve as a biomimetic structure. And lastly, we have the application itself, where you take the material and make use of that for various purposes. And that could range from disease modeling to study disease processes to in vitro safety and efficacy testing, or in vivo tissue transplantation. Now, in the interest of time, I'll skip this slide, which you can always refer back in the recording. So this is an example that shows how this workflow would be if you were to do a cardiac patch engineering. Let's move on to the applications. Now that I mentioned cardiac patch engineering, this is just one of the many downstream applications possible with bioprinting. As I mentioned before, it's a platform technology, so it can be used for many different applications, many different organ tissue types, and many different disciplines can use this. So I can summarize that the use of bioprinted products into kind of three broad areas of application. One is for toxicology research and drug screening, or for novel products, whether it be drug, cosmetic, or food. And the other one is the application as an in vitro study model for human tissue physiology and disease modeling, as in case of, say, cancer. And lastly, the Holy Grail application, which is being able to regenerate organs that are complex heterogeneous structures. So this would be for in vivo models for transplantation and regeneration. I don't want to go into the details of each of these, but you know what these applications are and how significant they are. So we will skip that, and just want to show you this video of how powerful the technique is in being able to recreate a beating organ like a cardiac tissue. All right, so in the context of all of these we have discussed so far about bioprinting, ROKIT Healthcare is emerging as a key player in bioprinting and organ regeneration space through innovative products, services, and robust in-house R&D programs, which are all aimed at creating breakthroughs in science and healthcare. ROKIT is dedicated to providing these innovative offerings to well over 25 countries around the world, with over 250 user institutions and partners. As a pioneering leader, ROKIT introduced the first state-of-the-art bioprinters that had the world's first clean chamber inbuilt into the bioprinter with the HEPA and UV lamps and hybrid print heads for both soft and hard tissue fabrication. That was the Dr. In Vivo 4D2. And more recently, as I showed you briefly, they launched the Dr. In Vivo 46, which is the world's first bioprinter and organ regeneration platform that has to fully integrate a cell incubator and plasma sterilizer, along with the 6 print heads for biofabrication, taking tissue engineering to a totally new level. I'll show you more about these features and capabilities again, if what I have shown before is not adequate towards the end of the talk. But now let's move on to some of the applications that some of our ROKIT in vivo users have put the system to use for. So because we don't have much time, I will touch upon these, and the goal is to simply give you a diversity of applications possible so that it inspires you to think about bioprinting as a potential solution for some of your own research. But we can always get you the references, and we can go back and discuss at length after the talk if any particular application is of great interest to you. So what I learned from Isha is that there is a lot of interest in this audience for the craniofacial and dental regeneration. So we will first start with the bioprinting applications in craniofacial and dental regeneration. The craniofacial complex, as we all know, is composed of components such as blood vessels, nerves, and also a variety of specialized tissues, such as bones, cartilages, muscles, ligaments, teeth. So it's a lot of different variety in a very small space. Let's put it that way. So craniofacial defects, they influence the structure and function of the jaws, face, all that. But they also pose a psychosocial issue that are related to the aesthetics of the craniofacial complex. So any defect in the face has multi-dimensional effect associated. So the size and shape mismatching can cause various problems, things as fundamental as facial asymmetry, language pronunciation, to even impaired mastication. So even though many concepts and therapies for general tissue engineering can and have been used for craniofacial tissue regeneration, still the more sophisticated tissue engineering approaches that can accommodate the heterogeneous and uniquely complex 3D geometry of the tissues are in big demand. So in that context, this research that we'll summarize here in this slide uses bioprinting technology to produce a 3-dimensional dentine pulp complex with patient-specific shapes by inducing localized differentiation of human dental pulp stem cells within a single structure. So what they've done is a fibrin-based bio-ink composed of fibrinogen, gelatin, and hyaluronic acid and glycerol were designed for bioprinting with human dental pulp stem cells. The effects of the fibrinogen concentration within the bioink was investigated in terms of printability and the human dental pulp stem cell compatibility and differentiation. Polycaprolactone, which is a biothermoplastic polymer often used in implantable medical devices, was used to reproduce the overall 3D shape of the teeth based on a 3D computer-aided design of a human tooth. In the dentine pulp complex, the human dental pulp stem cells at the center of the teeth, which you can see here in these figures, they were present as mesenchymal stem cells, and in the outer region, they were present as differentiated odontoblastic layer. Now, how did they spatially regulate the differentiation? So to spatially regulate the differentiation of these dental pulp stem cells, bioinks of higher fibrinogen concentration and overall higher stiffness and lower diffusion coefficient were used to induce the active odontogenic differentiation and localized mineral deposition. So bioprinting was able to precisely control the stiffness and the diffusion coefficient, thereby influencing the differentiation outcome. Now, this is just one tiny sliver or one example among probably, if you went on a literature search, you're going to find many, many hundreds of publications about the application of bioprinting in dentistry and craniofacial defect scenario. So the idea here is to emphasize that bioprinting offers some clear advantages and is very powerful in helping this research workflow with some notable advantages. One, you find that the 3D bioprinting offers rapid free-form prototyping capabilities that allow for fast fabrication of living tissue constructs of diverse 3D topologies compared to the existing tissue engineering techniques. Second, you also see that it has an excellent ability to construct tissue models with a homogeneous cell distribution and uniform spacing. Now that gives an advantage of repeatability of the experimentation. Last but not the least, you also have that bioprinting provides exceptional versatility in cell positioning, whether it is vertically in full layers or a planar in different cell compartments. Regardless, bioprinting can handle both. So there are these crucial factors to study a complex biochemical interaction or architectural complexity in living systems, and I think bioprinting can size up to that challenge. All right, so outside of dentistry, there are numerous examples of bioprinting research. So I'll quickly give a snapshot of the diversity, so you have some appreciation of that. Now, though these may not be directly related to the craniofacial engineering or dentistry, but exposing yourself to these examples will definitely give you great inspirations for deciding on the choice of bioinks or cells or 3D designs for your own workflow, for your own applications within the craniofacial or dental space. All right, so let's start with this first one here, where this is an example of developing novel biomaterial with bioprinting. In this study, which was done in Hanyang University of Korea, a group developed a synthetic self-healing hydrogel. They call it OHA-GC80H. Let's not worry about the name for now, but what they've done is they made this from modified forms of hyaluronate and chitosan. And a great thing about this hydrogel is that it forms bonds that are instantaneously without post-gelation or additional cross-linking processes. So what happens is after you cut these into pieces, as you can see here, the pink and the white, the two colored discs made this self-healing hydrogel reassemble and integrate completely in less than 10 minutes at room temperature. So it supported not only viability, but functionality of chondrocytes expressing the SOX9, as seen here in the gene expression. And it served, and going forward, what the group believes is that this self-healing hydrogel has the potential in cartilage, as well as various tissue engineering applications, and even in time-dependent drug delivery systems. This is an example of the application in adipose tissue engineering using bioprinting. See, conventional medical grade materials such as PCL are often used in breast reconstruction and nose reconstruction. But the problem is they do not match the mechanical property to the adipose tissue. So what this group did is they first developed a novel biomaterial called PLCL, and they mixed it with adipose derived ECM. Using the bioprinting technology, the group created a size customized scaffold, which when implanted into mice, it promoted adipose tissue regeneration with adequate macrophage infiltration and vascularization. The group confirms that the bioprinting combined with this new material and ECM can offer a new solution for burn, trauma injury, or post-mastectomy repair, and especially with applications in large defects. Here we've got an example of application in creation of salivary gland or branching epithelial tissues. And they call this as the nature-inspired catechol conjugated hyaluronic acid environment, or NICE, for boosting the salivary gland tissue engineering efficacy of the previously used biomaterials. So long story short, they combined this hydrogel coating technology with 3D printed porous PCL scaffolds to demonstrate that the highest amount of salivary gland tissues were formed compared to a non-coated ones. Or the ones that were using the traditional membranes of soft agar gels or agarose gels. So the group believes that this can also be applied for vascular endothelial cell growth, and they look forward to applying this platform to create better human models of other branching epithelial organs. All right, so now the next set of 3 slides will talk more about the cardiovascular tissues or beating models. So this one is about creating in vitro models for human pathological cardiac tissues using bio-artificial scaffolds. Here the group uses different patterns of PCL scaffolds to create tissues that mimic different electromechanical properties, and therefore they represent different stages in the progression of ischemic heart disease. And in this case, they used the bioprinter, and they used the iPSC-derived cardiac spheroids composed of fibroblasts and endothelial cells. And they combined them into multi-cell type spheroids, which are then mixed with alginate inks to form a bioink. The ink is then printed onto a hydrogel patch to create a model for the vascularized heart patch. This is work done in collaboration with the Fraunhofer Institute in Germany, and they developed a patented disc that allows iPSCs. The in vivo 4D bioprinter was used, and they developed a patent disc that allows the iPSCs differentiated into cardiomyocytes to align and create a beating model. It's very important to create a well-aligned tissue structure for the beating model to kind of be able to have the same electrochemical properties of cardiac tissue. And that's not an easy task, but using the bioprinting approach and our in vivo bioprinter, that was accomplished by this group. And as you can see, this beating heart has been beating post-printing for more than 6 months. So clearly they are quite viable as well. All right, this is a case of application of the in vivo bioprinter for the creation of muscle tissues. Cell suspensions of muscle tissues were printed around 2 PCL pillars, polycaprolactone pillars, to create muscle models for drug testing. The idea was to develop a 3D in vitro model to mimic human muscle tissues, and they used C2C12 cells in Matrigel. Okay, so in cancer therapy development, this study talks about cancer studies. Now, in cancer therapy development, understanding the complex and three-dimensional tumor microenvironment is very important. In fact, cancer was one of the very first fields of science to demonstrate that the 2D cell culture cannot quite replicate what actually happens in the human body. So, in this study, what you're seeing is this was jointly conducted by Junong University of Korea and Wake Forest Regenerative Medicine Center in North Carolina. The in vivo bioprinter was used to develop a 3D human bladder cancer model. It was compared with a 2D model to evaluate the behavior of the two types of cancer lines to two types of chemotherapies, one with rapamycin and BCG. Now, as one would expect, the results of the chemotherapy test showed different profiles of reactions with the cancer cell lines. So the group concludes that using 3D model of cancer is very important to better assess drug efficacy. So lately, more than ever before, bioprinting is being explored to create disease models that can better predict drug responses. And cancer, as I mentioned just now, is one of the hottest topics in this area. So as we all know, existing in vitro models of cancer are often single cell spheroids, and they lack the tumor microenvironments crucial to their intrinsic behaviors and malignancy. So here in this example, what you're seeing is we've got a bioprinted layer of lung fibroblasts and then human vascular endothelial cells in bioinks of alginate, fibrinogen, and gelatin. The multicellular tumor spheroids of glioblastoma cells are seeded onto the vascularized tissue and incubated, resulting in vascularized tumor models. And okay, this is a bone implant study. As we know, the autologous bone grafts are regarded as the gold standard for restoring bone defects. However, for large bone defects, large dimensional bone defects, there is a need for alternative methods to autologous bone grafting, which, although effective, they can cause other new problems in the donor site, such as morbidity and low graft availability and so on. So for this reason, bioprinting has been explored quite actively to fabricate bone regenerative scaffolds using synthetic bone materials, among which hydroxyapatite is most common used material. Now, in this particular study, what you're seeing here, bioprinting was used to develop an entire customized bone implant manufacturing process in GMP using hydroxyapatite, PLLA, beta-TCP, and it received the approval of for distribution by the Korean FDA. So this is a good example of how commercialization is possible. And bone implants not only would be using this approach would be applicable for orthopedics, but they can also be used in dentistry for maxillofacial implants as well. Okay, this is another study with respect to bone regeneration. So in short, what's been done here is a strontium has been incorporated to create a bioceramic scaffold, strontium incorporated into hydroxyapatite to promote cell proliferation, osteogenic differentiation, and cellular mineralization, and can be thus used as a novel bone substitute. And here we have a study that's done in collaboration with Mass General Hospital in Boston between ROKIT and MGH. And this is currently conducting a preclinical testing for cartilage regeneration using stem cell laden bioprinted matrices. So in the cartilage defect models in beagles, as you can see here, the results so far have been quite encouraging, and quantitatively, we have seen that the printed hyaline cartilage, the localized hyaline cartilage regeneration happened in the knee, and qualitatively, the beagles were observed to have recovery of walking functions. There is a lot more to go. So this is, I'll quickly skip through this in the interest of time. So again, you've got the applications in nerve guidance conduit production to align collagen hydrogels and enable using 3D printing patterns to provide a preferable environment for nerve regeneration and providing a guidance path for the nerve to propagate. Here we have other applications outside of the organ or tissues, and this is a particular case where in vivo bioprinter was used to prepare a QR-coded drug delivery system in the form of a oro-dispersible film. And these have the potential to prevent misconsumption, misadministration of medicine, and also they could replace anti-cancer injections with oro-dispersible films, which would be more effective and minimally invasive. Lastly, we have got an example of use of bioprinting to develop an egg-shaped bioresponsive device that combines microbes, hydrogels, and conductive nanofibers. The reason why I talk about this is, so it's not always cells that we have to use. So your ability to apply bioprinting is only limited by your imagination. The applications are numerous, and a combination of your imagination and the power of the bioprinting can lead to unexpected and fascinating outcomes. And this is one example to demonstrate that. But of course, all of these that we have discussed so far are mostly applications in the research domain. So the real excitement comes when we are able to take these applications, put bioprinting from the bench to the bedside, and create autologous treatment solutions for patients. So with that in mind, as I mentioned before, ROKIT Healthcare has been actively pursuing the diabetic foot ulcer treatment project. So they've been translating the benefits of automation and personalized therapy using bioprinting. And in the last 2 years, it has had the most achievements in creating an application in diabetic foot ulcer area. As we all know, diabetic foot ulcers are chronic conditions, and the side effects affecting almost 25% of all diabetic patients at some point of time in their life. So for more than 20 years, the traditional wound care systems have relied on dressings and negative pressure wound therapies and collagen patches and autografts, but there was never really a truly autologous regenerative strategy employed. Now, using the Dr. In vivo bioprinter system, ROKIT Healthcare has developed a bioprinting-based skin regeneration process in which the bioinks are the ECM and the microfat particles obtained from patients. Now, these bioinks are printed into specific widths and custom made for the wound. So far, ROKIT has spearheaded the clinical studies in India, Korea, USA, and Turkey, and 40 patients out of 40 controls ended up recovering well. And here is just examples of the ROKIT Healthcare's clinical study result in India, which is a country especially hard hit by the diabetic foot ulcer. The results have been extremely encouraging, and it took, on an average, 4 weeks to close the wounds that had otherwise stayed open for months or even years. Think about that. So we envision this, that this therapy using the bioprinting approach can be applied to more skin conditions ranging from burns, scars to even bed sores. And as an extension of this idea, one can imagine that therapy can eventually be called as an integrated 3D scan print scan print therapy by taking the patient image, and it translates into personalized regenerative solution at the bedside. So this video might be quite entertaining to watch. As we can see here, I'll just quickly glance through this video. So it's possible to take the wound, scan it, and then digitalize the wound area, and then customize the printable structure to fit the specific wound, and then be able to actually fabricate the wound and have it printed with patient's own patient-derived material. So all of these would be like a minimal manipulation. So can be done right in the hospital operation theater setting, and apply it back to the patient. And that could result in very promising therapeutic solutions. All right, so that concludes the applications that I wanted to cover today. Again, this is just the tip of the iceberg. Now, there is so much out there, and there is so much possible by employing a robust system like ROKIT in vivo bioprinting platform, and your imagination and expertise, and your deep knowledge of your own systems, and your specific problem areas and workflows. We do have an extensive network of existing users from across the globe, both US as well as many other parts, as you can see here. And as I just started talking in the beginning, we do have two bioprinting platforms available. One is the in vivo 4D2, which is a dual module, more like the early first generation. And then we've got the 46. So I think I have already gone over this, so we can revisit this towards the end. But in conclusion, what I want to say is that if we were to talk about the essence or the take-home message for today's talk is that the bioprinting brings three breakthrough benefits to research and novel therapy developments. One is automation, making it faster and precise. Then we've got the physiological relevance, and then we've got the on-demand fabrication. Now, what all these three entail is the ability to experiment with better repeatability and better physiological relevance, the ability to create biomimetic structures and tissues without having to depend on model systems that are either expensive or simply not ideal. And then the ability to create more sustainable research methods and completely phasing out animal models at some point in future with alternative human cell-based methods of testing. And it's possible for us to imagine that the bioprinting facilities could be set up in every region of the world using this technology, and that would have the potential to completely transform regional biomanufacturing value chains, just as COVID-19 gave us a hint of its needs. And it won't be surprising for you to realize that actually this idea is already happening as we speak, because there are indeed centers of bioprinting excellence that have been established at national and international levels over the last few years to pursue this. And I even heard that NASA is heavily invested into this technology, and we do have bioprinters up in the International Space Station to pursue solutions for space exploration and manned missions in space. So the last point I want to make here is that bioprinting is a very powerful technology. Now, we are living in an era of regenerative medicine, and in this era, bioprinting is not only a revolution that's underway for the way we create and use model systems for answering basic biological questions. It is actually revolutionary in the way you even practice medicine, because instead of using systems and methods that rely on treatments of symptoms using small molecules or monotherapies, what bioprinting is doing and trying to do is to shift the focus towards harnessing body's own healing potential inherent in these stem cells. And this is also the vision and mission of ROKIT as a company. The future of medicine is already being realized today by these, by those pioneering, by the people who are pioneering these technologies in various disciplines of science. And dental and bone applications are one of the actually early wave of applications that we will see. So with that, I'd like to thank you all for sharing your interest, passion, and willingness for innovation in this area by participating in this seminar today and giving me and my team the opportunity to present. I'm very encouraged by your current experience and interest, and I hope you will be able to adopt this for your own labs, and hopefully we can get you a ROKIT in vivo bioprinter very soon. Thank you. Sorry if I went a little overboard on time. Everything changed since our going from these slides backwards.
Yeah, I'm sorry about that. I was the
No problem,
person.
but that's what we wanted. Usually we end up talking and then we run out of time. But thanks for the patience.
Thank you so much for the presentation. It's quite enlightening how much the field has advanced over time. Like there's a lot happening everywhere.
Oh, yes.
With the realization that the conventional techniques no longer hold the premise.
Right. And I think there is so much activity even within ROKIT that amazes me. They have so many different projects, and Dai and we have Hanjun, I guess, who can also talk more. In fact, he can talk more about the DFU if you still have questions on the skin printing.
Okay.
But please ask any questions that you or your team might have.
I'll let others talk. I think I've done enough talking for the day. I'm unsure if anybody is gonna even ask. Does anyone has any questions for Senthil and his team? Sure.
Hello?
Normally shy.
Yes, hello? Hello?
Yeah, Tamir, go on.
Yes, I just have some questions. So, what is the printing resolution that can use for that technology? And is there a famous 3D scanning technology for example, for the heart cell or tooth cell? That can give you the high resolution information for the tissue, for the heart tissue or tooth tissue.
Sure. Yeah. Typically, when you're printing cells, first I'll offer my thoughts, and then I'll let my colleagues chime in on anything that I'm missing out. You don't want to squeeze the mammalian cells below 100 microns or so, because they just will not be happy. So when you're using cell printing, 100 microns to 200, normally 200 microns is what a lot of people use, but 100 to 200 is a good nozzle size to use. So that kind of limits the resolution of cellular structures. But if you're using for scaffolds, you can go down to 30 microns or 80 microns, somewhere in that range. And in terms of the resolution of the printing itself, depending on the technique, most micro extrusion techniques can go anywhere from 5 microns to 1 millimeter range. And laser assisted and those types of techniques can go from 100 to 600 microns, or inkjet can go to 50 microns and so on. But either way, micro extrusion is more compatible with cellular printing. In terms of what was the resolution for the cardiac tissues, I can't answer that right now, unless Dae or Kim, if you have any inputs there.
I think, well, if I understood his second question correctly, I think he was asking about if we know of any high resolution 3D scanning technologies for cardiac tissues. So at this point, I do not have insights into specific brands or products of 3D scanning technologies that may be fitted to your work, but maybe I could look into what groups or suppliers could provide better information for you. Is there a particular aspect that you are looking for?
No. I think it's a general question. It's not my interest, but it's just a general question. Thank you. Yes.
Okay. Yeah. In addition to what Senthil said about the nozzle size dictating the resolution of printing, if you are also talking about the resolution of the distances between the lines that you end up printing, that would be dictated by the motor resolution of your bioprinter. So that would basically depend on what motor you use and the steppers inside it. So for the bioprinting platforms that we have developed, the motor resolution can range from 50 microns to 10 microns, depending on 42 versus 46, which is the higher resolution with the medical grade linear robot system built inside. Does that answer the question?
Yes, thank you. Yeah.
Does anyone else has any questions?
I actually had a question for Isha.
Okay. Normally it goes the other way around. Doesn't happen this way.
2 ways.
No, go ahead. Sorry.
So I understand that the groups that are in this meeting today mostly belong to the Department of Dental Medicine, Craniofacial, Restorative Engineering, but you expressed some interest in the diabetic foot ulcer treatment platform that we briefly introduced. Is the reason that you show interest perhaps related to the treatment similarities between like oral mucosal tissues versus skin tissues?
Kind of. As I mentioned, I'm collaborating with Jacob Toller's lab on trying to find a way if we can actually use cell therapy for treating RDEB. They've actually shown it, that they have used stem cell therapy to treat the condition, but we're trying to find ways how to apply it clinically more effectively. So it's actually for the skin regeneration in that space. And me being based in the dental school, we do have projects which involve either the soft tissue regeneration, which can be either skin regeneration on the osseointegrated orthopedic implants, and also your gingival attachment to the dental implants. So I was just curious to know what were your thoughts, or what did you feel with the diabetic foot ulcer treatment? What was the makeup of the ink? And that showed promise that you're actually seeing significant improvement in wound healing. So I was just curious to know about the composition and stuff like that, just to have a better understanding of what's your take on the system.
Mm-hmm.
Yeah.
So we actually have Mr. Hanjun Kim, who is the
Hello.
Hello.
Isha and Santo. Yeah, I was trying to listen just to and also Daya has more experience in the bioprinters. I'm quite new to the company, but I'm sitting in I'm representing diabetic foot ulcers strategic business unit as a principal director here. So yeah, just I'm more focused on the hospital applications, and this is basically using the fat tissue through the liposuction, and you're using your own fat tissue, and we're basically doing the sample preparations and mounting the samples which is contained in the dispenser, and we are basically using our just own fat tissue and making a biopass and applying to the wound area. So not there isn't any we do this within minimal manipulation. There is nothing that has any additives that has any chemical reactions or anything like that. Yeah, I don't know whether that answers your question.
It does kind of, but how?
Hello?
I think she's frozen.
Okay.
Can you hear me now?
We lost you there.
Yeah.
Go ahead.
Sorry, I don't know what happened right there. Yeah, I do understand what you said about minimum manipulation of the ECM, but how do you ensure that this patch that you have developed is stable even during the first few hours of when you do the surgery or when it's applied to the patient? And was my understanding correct that you actually use cells as well? And if that's the case, won't you get variability in how many cells you get from different patients? So that won't that add another variable when you're treating different patients? Because you're not manipulating it, you're not expanding it, you're just derivatizing it and using it straight away, right?
Right. So it's an ECM as extracellular matrix, but like we don't really concentrate on the cell concept. Yeah. So we are basically implanting the tissue itself, and then that has all this all the components and growth factors and everything there, and we're just relocating those to the different area. Yeah.
So no external cell is added to the mix. It's just the ECM patch.
Right.
Okay.
That's right. Yeah.
Got it. Thank you.
And by virtue of being adipose isolate, they do have some stem cells in the mix.
That
by default.
That is what I thought, but you're not calibrating the system anyhow, right? So you would still be getting patient to patient variability. Depending on how. Yeah. So do you have any take on addressing those issues to make sure that your system is relevant? The patient's age would make a difference on how multipotent the stem cells are and how much regenerative capacity they have. No.
Do you have any? Would you like to add anything here?
I think based on our experiences interacting with doctors and healthcare professionals, it was necessary to basically modify the volumes of the liposuction volumes, or the stromal vascular fraction from the adipose derived tissues that we are using, specific to the patient or specific to the wound defect. But outside of that, we have basically assessed the minimal, what would you say, the sort of the minimal amount of the SVF that is required to catalyze or to accelerate the regeneration, the endogenous tissue regeneration in these types of chronic diabetic foot ulcers. And we are sort of abiding by that point, plus alpha, according to the patient demographics. So I do not have, like specific numbers that would be matching different patient groups, but we do have data basically showing that this method that relies on autologous solutions from the patient basically accelerates the epidermal healing that in turn minimizes a potential infection or other, sort of other things that could cause the worsening of the symptoms. And so that is complemented with other, of course, other combination therapies such as medicines or negative pressure wound therapies in order to overall improve the outcomes. So we are currently working with David Armstrong from USC, who is a renowned podiatrist and a diabetic foot ulcer surgeon for the last, I believe, 30 years or so. So if we have more questions about that, we can definitely, give more inputs by connecting you with the sort of the clinical teams in-house. But yeah, I hope that Mr. Hanjun and our answers were able to address some of the questions that you are wondering about.
Yep, no, that's good. And if I have any questions, I can get back to you on that.
Yeah, sure.
Is doable. No, that's good. Thank you. I don't think anybody else wants to ask any questions. Okay, thank you so much. And if we can have access to the recording, that would be great, at least for Alex and Dr. Chu, who couldn't make it to the meeting today. So
yeah, sure. We would be happy to share the recording afterwards. And if you guys have any questions, feel free to email us as well. And we will keep in touch.
Thank you so much. Really appreciate that. It's pretty exciting.
Yeah. Senthil, you can make closing comment. I think you're on mute right now.
Sorry. Yeah, I'm sorry. All right. Thank you, everyone, for your time and patience today. Isha, just wanted to mention to you, if you or anybody in your group has any ideas for a bioprinting collaboration or with ROKIT's R&D, or if you have needs for any biomaterials, especially the ECMs, that's also something we would love to hear about. And of course, we would like to see our in vivo 46 platform as part of your research equipment whenever that's possible.
Okay.
So please let me know.
Okay. If you could share the information that you previously mentioned regarding the ECM,
yes,
that would be great, because I would be interested in looking more into that aspect.
Okay. All right. I'll send you the Humatine flyer.
Okay. Thank you.
Thank you all. Thank you again. Have a nice day.
You too. Thanks. Have a good rest of your week.
Thank you all.
Have a good rest of the day.
You too.